I wanted to thank the conference organizers for giving me this chance to talk a little bit about some of the work we've been doing in our lab on developing model systems, 3D organoid models of lymphatic malformations. So briefly, just as a point of introduction, the lymphatics are a series of vessels that have these highly specialized contractile vessels that serve to transport fluid from the interstitium back into the circulation. As you can see from these two videos here from our lab, one of an isolated vessel taken out of the body on the right, and then another of a vessel imaged in vivo. Now, lymphatic malformations are a particular disease of lymphatic vessels. It's an overgrowth syndrome in which the endothelial cells have a mutation in the PIK3CA gene in most cases that causes hyperproliferation and overgrowth of lymphatic tissue. The clinical presentation and disease severity can be highly variable depending upon the location of the malformation and perhaps when the mutation is acquired during development, as is shown here, some different examples. So I mentioned about probably 70% of malformations are due to mutations in PIK3CA, and in here there was a study that sort of took a sample of patients from this clinic in Seattle and found 3 different types of mutations in PIK3CA that comprise the majority of them. Because PIK3CA has been identified, people have been using sirolimus off-label for a few years now as an inhibitor of mTOR since the hyperactivation of AKT is implicated due to this mutation. And so this has been used off-label, and it's proven to be rather effective in many patients in sort of controlling the growth of the tissue and even reducing its size. There's interest in other more specific inhibitors, and also trying to understand why patients respond differentially. In vitro models of the disease could be quite useful in sort of understanding the mechanism, as well as exploring new therapeutic options. And so there are a variety of models that people have used in vitro. So sort of the most straightforward is there have been groups that have isolated endothelial cells from lymphatic malformations and shown that if you plate these and expand them, they do exhibit hyperphosphorylation of AKT pathways, and then they are more proliferative. And in this paper here on the right, you could use things like rapamycin or other PI3K inhibitors to show responsiveness of those drugs in vitro. Now, in addition to these models, people have also used Xenograph models in which you take these similar purified cells from patients and then put them in an immunocompromised mouse, where you get sort of the incorporation of the into more of a tissue-like structure that mimics sort of the disease phenotype. Or excitingly, there's even a genetic model of lymphatic malformations that was recently published in which, in this case here, a specific PIK3CA mutation that's responsible for some disease cases was driven by a VEGFR3 promoter, so that you had lymphatic-specific overactivation of the PIK3CA signaling pathway, and this was a Taya Mekkinen's group that published this work. So we've been interested sort of trying to bridge the gap between sort of the kind of simplified in vitro models and then in vivo models. And one of the ways we do this is we engineer biomaterials to support tissue or cell growth and create these so-called tissue organoids. And so a platform we've been working with is a PEG-based hydrogel, PEG-formol. The advantage of this is it's highly tunable, so we can control like how the degree of cross-linking, and so how stiff the gel is. We can control its degradability. We can control presentation of growth factors, peptides for matrix attachment, and sites for cell adhesion. And so Josh Hooks, who was a former Ph.D. student in the lab, he took this platform and engineered it. And the first thing he was interested in doing was targeting the tissue stiffness of native lymphatic tissue. And there was this one publication by Mika Fry when she was in Taya's lab, where she measured the stiffness right outside the cardinal vein, and this paper suggested stiffness could even be a cue to promote lymphatic migration, and that lymphatics prefer very soft tissues, and so we can make hydrogels right in this range of tissue stiffness. And so Josh went on, and I'm not going to show all the data, but he tested a variety of different conditions, stiffnesses, peptides, degradability, and sort of optimized this hydrogel to support sprouting. And so this was done with rat lymphatic tissue. So basically took collecting lymphatic vessels from the rat out and cut them into segments and embedded them in these hydrogels. And here in example C is this 10% PEG with 2 millimolar RGD, fully degradable hydrogel that supported complex branching. So Josh went, and then he took this hydrogel platform, and in collaboration with Matt Hawkins, who's at the Children's Healthcare of Atlanta Children's Hospital and the director of the Vascular Anomalies Clinic there, Matt was able to secure us patient tissue from patients undergoing sclerotherapy. And so we get a biopsy of the malformation from the patient, bring that biopsy back to the lab, and cut it into little pieces, but preserving the native microarchitecture, and then embed it in the hydrogel. And this is an example of one such biopsy that's been placed in a hydrogel and grown for 10 days. And so, 90+% of what you're seeing here is new tissue growth in the hydrogel after the explant is placed in the gel. So Josh went on, and we made these disease organoids from 5 different patients and did a dose response to rapamycin. And what he found was in this patient cohort, the 2 patients that happened to be from a macrocystic or mixed lesions showed a dose response effective rapamycin, whereas the 3 patients that were from the microcystic were not very sensitive, did not respond to rapamycin treatment. Josh also went on, and he took a rat collecting lymphatic tissue, which would have no hyperactivation of the PI3K pathway, and he showed that rapamycin had no effect there on the sprouting in these concentrations. So the advantage of this approach is we take the intact tissue directly from the malformation and grow it. Of course, the disadvantage is you're really limited on the number of these organoids you can create from a single patient. And so we were interested to see if we could take endothelial cells that have been potentially take endothelial cells derived from lymphatic malformation patients and reassemble them, because then this would allow you to expand a library of cells up, and then you could do really, much unlimited number of experiments, at least out to 8, 9, 10 passages of cells where you could create these cellular aggregates. And so for proof of principle, we started using rat endothelial. So we took rat lymphatic endothelial cell lines and rat lymphatic muscle cell lines and optimized a formulation where we take the 2 cell types, we coat the cells in matrix proteins. They self-assemble into spheroids, and then you can actually see them here in suspension. And then we put those spheroids into our hydrogel, and they form remarkably similar networks. You can kind of make out the original body here as direct lymphatic vessel culture did. And so we found that they're responsive to stiffness and they're responsive to growth factors. And interestingly, we found that and this isn't a large stiffness range, but really soft hydrogels and then really stiff hydrogels don't sprout on their own very well, but when you supplement them with VEGF-C, then you can get sprouting, where in this intermediate range of sprouting, the addition of VEGF-C over what's already, the growth factor is already present, 10% serum, didn't really have an additive effect in promoting sprouting. And that's just quantified here. Again, we see, so particularly soft hydrogels are very responsive to growth factors. So I think this motivates the fact that even the tissue microenvironment can control the phenotype and progression of lymphangiogenesis and the responsiveness to growth factors, which is an area we're interested in exploring in lymphatic malformations. although I'll say all that data there was from rat cell endothelial cells. So in summary, I think we have these two platforms, enabling platforms where we can take tissue. We can either, if we have a cell lines isolated and expanded from that tissue, we can self-assemble them and grow them into the hydrogel, or we can directly take intact tissue and grow them and use them for purposes of understanding mechanism of drug resistance, understanding what the relationship between the mutation and responsiveness to therapy. And so with that, I'd like to thank the funding sources that got this work started. Josh, who really developed the initial hydrogel platform, and Darlis, who's a current Ph.D. student who's working on these spheroid body approaches. With that, I'm happy to take any questions.